Hi everyone, welcome to another lecture for Anatomy and Physiology 1. In today's lecture, we're going to be looking at the muscular system. More specifically, we'll look at the different muscle types that exist in the human body. We'll look at how muscles actually work, so down at the physiologic cellular level. We're going to look at reflexes and the energy that's required in order for muscles to contract. We'll start by looking at the different types of muscle tissue that exist. We have skeletal, smooth, and cardiac muscle. Skeletal muscle is considered to be striated, and so is cardiac muscle. And when we say striated, we're meaning that it possesses these bands that give it a groove-like appearance. So those are found in both skeletal and cardiac muscle, and so we can sometimes call them striated muscle. And that differentiates them from smooth muscle, which does not have the striated appearance. Skeletal muscle cells are voluntary. This means that you have control over the muscle contraction. Now remember that when we talk about muscle cells, we can call them muscle fibers. And skeletal muscle possesses these thread-like cells that have, as I already mentioned, many of those striations, which we can call cross striations. There's also many nuclei found per cell, and you can see the groupings of nuclei found here. Smooth muscle, on the other hand, which we know is non-striated, and that's where it gets its name smooth, because when we look at it microscopically, it has a smoother looking appearance in comparison to the other muscle types. Smooth muscle is involuntary. This means you do not have any control over its contraction, and this is the type of muscle tissue that we would find in, for example, your stomach. It's found as part of blood vessels, your intestines, and many other structures within the body. And so these organs that have smooth muscle are able to contract, but again, you don't have control over it. So if you think of food moving through your intestinal tract, if you think of your stomach, which is breaking down food physically, if you think about the movement of blood through your blood vessels, it can move in one direction because of the presence of muscles that contract, particularly those smooth muscles. The cells of smooth muscle are elongated, quite narrow cells. They don't contain the striations, as already mentioned, and there is one nucleus present per cell. Cardiac muscle is only found in the heart. That's the only location we'll find this type of muscle tissue, and it is also involuntary, which makes sense because you don't have control over your heart beating, at least not where you can tell your heart to contract, but of course it will respond to certain situations such as stress and excitement and exercise. But remember, you're not telling your heart to beat slower or faster, it's just responding to a situation. Cardiac muscle possesses branching cells that have these intercalated discs. So intercalated discs would be made of, thinking back to the start of the semester, gap junctions as well as desmosomes. And the possession of intercalated discs is going to allow for the nerve impulse to be transmitted very easily, that communication from cell to cell throughout your heart. On this slide, we're going to look at some of the similarities and differences when we compare cardiac muscle to skeletal muscle, because they do have some similarities, but then some big differences as well. So when we look at the similarities, both contain parallel myofibrils, T-tubules, and a sarcoplasmic reticulum. So let's elaborate on that a bit more. And this here is cardiac. These are cardiac cells that I'm showing you here, and you can see the branching cells that I spoke of earlier. You can also see those intercalated discs shown differently depending if we're looking at the outside of the cell versus the inside of the cell. And remember, those contain both desmosomes, but also gap junctions. So the similarity in terms of parallel myofibrils. Remember from our lecture discussing muscle fibers, that this would be a muscle fiber, this is a muscle fiber, and this is a muscle fiber. What we hadn't gone into talking about yet was what's within the cell itself. So the muscle fiber, what's within that muscle fiber. And so that's what today's lecture is going to be focusing on. And one of the things found within muscle fibers is myofibrils. Myo means muscle, fibrils are like small fibers. And so these here are representing myofibrils. And so myofibrils then are a component of the muscle cell or the muscle fiber of both cardiac and skeletal muscle. We also have T tubules found in skeletal and cardiac muscle. The T stands for transverse, and this will be discussed in more detail soon. But as an introduction, we have these transverse tubules that extend transversely. It's like a tube-like structure. And the purpose of this, as we'll see, is going to be allowing electrical signals or impulses to really traverse or travel along throughout the cell, so it can get deeper into the cell. And then we also have a sarcoplasmic reticulum. The sarcoplasmic reticulum is really a muscle fibers version of the smooth endoplasmic reticulum that we've talked about in the past. And the purpose of the sarcoplasmic reticulum, as you'll see later, is to store calcium ions until they're needed. And we'll see the importance of calcium ions with contraction later today too. Now, in looking specifically at features of cardiac muscle that distinguish it from other muscle types, we know that it's found only in the heart, and it's going to form the walls of each of the heart's 4 chambers. And we'll learn about this in the cardiovascular unit, but the chambers are going to contain blood that will be pumped by cardiac muscle out of the heart to areas such as the lungs and the rest of the body. Cardiac muscle fibers form this continuous contractile band around the heart chambers, and they will conduct a single impulse that will travel all throughout the heart. And that's what makes it unique. And remember, because it has those intercalated discs, those gap junctions that allow for the impulse to be transmitted very fast throughout the heart. And the way that the heart is designed, it's almost like it's just a single plasma membrane across all of the cells. And a plasma membrane specific to a muscle cell is called a sarcolemma, and that's another term we'll learn about later. We know each muscle fiber is typically branched with cardiac muscle. The T tubules, those transverse tubules, are larger in cardiac muscle, and they form dyads. So what this means, what a dyad is it is a T, for every T tubule, we have one sarcoplasmic reticulum that's associated with it. So di means 2. Whereas with skeletal muscle, it forms a triad where it has one T tubule and 2 sarcoplasmic reticulum. And finally, cardiac muscle really forms what's called a synctium, which means that they can conduct electrical activity from one cell to another without having to have individual nerves that supply, or we say innervate, each part. And this allows for, again, real fast transmission throughout the heart, which is the goal. On this slide, we can see some additional comparisons of cardiac and skeletal muscle, but we're looking at more of the function or the physiology. Starting with this graph here, this is comparing the actual contractions. And what happens with skeletal muscle is you end up with a brief nerve impulse, and that's going to trigger a brief twitch contraction. So we have the brief nerve impulse. So that's the stimulation of the muscle and the brief skeletal muscle contraction. But what you get with cardiac tissue or cardiac heart muscle is you're going to get this more prolonged impulse in the heart tissue. Okay, you can see that here, which is going to then produce a rather slow and more drawn out contraction. So let's look at this chart here, comparing some features in terms of physiology of muscle. The length of impulse we already spoke about. With skeletal muscle, it is a short and quick impulse, whereas with cardiac muscle, it is a longer, more sustained impulse. And the feature of a of skeletal muscle, where it has that very short, quick muscle impulse, it can allow it to lead to tetanus. And in physiology, tetanus is the prolonged contraction of a muscle caused by rapid, repeated stimuli. So we can have rapid repeats of this impulse to create tetanus. Whereas with cardiac muscle, we won't get that because the impulses aren't occurring quick enough to produce tetanus. In terms of ATP supply, so this is our stored form of energy, it's going to be depleted very quickly in skeletal muscle with those rapid impulses that lead to contraction of the muscle. And because of that, our skeletal muscles are more likely to fatigue. And you've experienced that many times yourself, where you've overworked your muscles or you've worked them hard and then they fatigue. Whereas with cardiac muscle, we have a continuous production of ATP because we can't afford to have our heart fatigue. Now, in terms of how the muscles receive their stimulus in order to contract, with skeletal muscle, it will require a trigger from what's called the neuromuscular junction. And this is the junction between a nerve and a muscle, and that will be discussed in more detail in the nervous system lecture. But with cardiac muscle, it's self-stimulating, so it has its own ability within it to generate a stimulus or an impulse to send throughout the heart, and that will be talked about in the cardiovascular system unit. In terms of type of contraction with skeletal muscle, it can lead to fatigue, which we know with cardiac muscle, it's going to contract in a very rhythmic and continuous way so that it can maintain constant blood flow throughout your body for survival. We'll now compare smooth muscle to skeletal muscle, looking at similarities and quite a few differences. There are a number of terms on this slide that won't make sense to you right now, but the terms will be defined throughout the rest of the lecture. And so once you review the lecture, which I highly recommend you do shortly after watching this lecture, a lot of it will make more sense. So in terms of similarities, both skeletal and smooth muscle contain thin and thick filaments that make it up. And this is just showing you here the thin and thick filaments that will contribute to the contraction. But we'll come back to that. In terms of differences, first of all, even though they both contain thick and thin filaments, they're arranged differently. In skeletal muscle, they're arranged in a way that they end up forming those striations or those bands that you would see when you look at it microscopically. But with smooth muscle, they're arranged differently, and so much so that they give a smooth appearance, which differs from the striated muscle in skeletal and cardiac muscle. In terms of the design of the muscle, these are shaped a little bit differently. They're smaller than our other muscle cells, and they're tapered at each end, and then we already talked about there being a single nuclei. There are no transverse tubules. There are no sarcomeres. The sarcomere is sort of an organization of some of the components of the muscle cell, but that will come up later. And the sarcoplasmic reticulum is arranged differently. It's very loosely organized. And remember that this is the muscle's version of an endoplasmic reticulum. As you will learn soon, in skeletal muscle, calcium is going to be stored in the sarcoplasmic reticulum, and then it's going to be released from there to bind to a protein that's called troponin. And this will cause a contraction. But smooth muscle cells are different in that instead, their calcium is going to be coming from outside the cell instead of inside the cell from that sarcoplasmic reticulum. It's going to come from outside the cell, and it's going to instead bind to calmodulin in order to trigger a contraction. And you can see in this image the appearance of a smooth muscle cell, or also known as a muscle fiber, in its relaxed state versus its contracted state. The rest of today's lecture will discuss skeletal muscle specifically and their contractions, but we're going to look at some of the characteristics and functions of skeletal muscle cells. So they possess excitability. So when they're stimulated by nerves, they will respond to it. They also possess contractility, so they have the ability to contract or shorten, and when they do, it produces movement of the body. They possess extensibility. So this means they have the ability to extend or stretch, which will allow the muscle to return to its normal resting relaxation length after the contraction has occurred. Muscles may also extend while still exerting force. So if you think of yourself with a heavy object and you're lowering the object down, the muscle is extending in that position or through that motion. In terms of function, skeletal muscle cells allow for movement, heat production, as well as posture. In terms of movement, we know muscles will pull on bones to create movement of that bone. Muscles can also pull on other muscles, but they can also pull on the skin, like what we spoke about with facial expressions. Our muscles can allow for us to move our body entirely or just certain areas of the body. But when we produce movement of the body as a whole, we call this locomotion. Muscle cells, like all cells, produce heat by the process known as catabolism, and being able to produce heat will contribute to homeostasis of temperature in our body. And finally, posture. So having this continued, what we can say, partial contraction of many skeletal muscles is going to make it possible for us to stand, sit. It's going to allow us to remain relatively stable while we're undergoing a task such as walking or running or performing other movements. We've already learned about skeletal muscle as a whole and what makes up skeletal muscle and the different coverings and components. But what we haven't looked at yet in detail is what each of these muscle fibers or muscle cells are composed of. And I made a few comments at the beginning of the lecture on a few of the components, but let's go into detail now. What we're going to do here is pull out a single muscle fiber, or what we call a single muscle cell. Remember, a fascicle here is a grouping of a bunch of muscle fibers or muscle cells. So we're just going to pull out one and see what makes it up. The first component are what we can see as being these blue and pink regions of the muscle fiber that are packed together. And these are fine fibers, and they're called myofibrils. And they're closely packed together, and they fill the sarcoplasm, which is the cytoplasm of a muscle cell. They extend lengthwise throughout the cell, which we can sort of appreciate here. It looks like a grouping of really small straws. And they contain myofilaments. And we'll talk about those coming up. So once again, myofibrils contain myofilaments. And we can say that a muscle fiber contains myofibrils, which contain myofilaments. I mentioned the term sarcomere before. A sarcomere is a feature of skeletal muscle. It's a segment of the myofibrils between two successive Z discs. So what does that mean? Well, we'll look at Z discs soon, but just to show you now, this is a Z disc and this is a Z disc. So from here to here, the region between two successive Z discs is called a sarcomere. And a sarcomere is very important because it is the contractile unit of the muscle fiber. So next up, we're going to look more closely at these myofilaments that we just spoke of. Okay, so always looking at your headings on your slides so you know what we're talking about. We're going to look at 4 different types of proteins that are part of myofilaments. Now, from our previous slide, we already know that a myofibril contains myofilaments, but it contains different myofilaments. More specifically, it contains some that are thick and some that are thick. We're going to start off by looking at the protein myosin. Myosin is what forms most of the thick filament, and it's made up of a myosin head, which we'll see here. So here's myosin, and here's the myosin head. You can see many heads, and they are going to be attracted to another type of protein that's called actin. So here is our actin here. Actin is made up of these two twisted fibrous strands of protein, and they will form the thin filament. Actin contains some active sites that are going to attract the myosin head. So in other words, the myosin head during contraction will bind to the active sites that are found on actin in order to produce the contraction. Two other important proteins include tropomyosin and troponin. Starting with tropomyosin, that is this sort of like thread-like structure that you can see here. And what it does, and you can kind of see it happening here, is it's blocking, when the muscle's not contracting, it's blocking those active sites because we don't want a muscle to contract when it's not supposed to contract. So it's blocking those active sites. Whereas troponin, this protein you can see here, troponin is going to hold that tropomyosin in place, keeping those active sites that would be underneath it covered. When this myosin head attaches to the active site, we'll see that it forms what's called a cross bridge. We're going to talk about the features of skeletal muscle when we look at it microscopically. So here would be a muscle fiber or muscle cell, which we know is composed of the myofibrils, both thick and thin filaments. We talked about actin and myosin. Here what you can see, what's represented in blue is our myosin, and what's in pink is our actin, so that looks similar to the previous slide. We also said that this is the Z disc from here to here, and that from one Z disc to another Z disc is called a sarcomere, and that's the contractile unit. So let's look at some of the other features. We have an area that's called the A band, shown here. So the A band is really from where the blue here starts, the myosin, to here. This is the A band, which is a dark band that you would see histologically. So it's cut off a bit here, but the A band would be here. So this is the dark stripes that you would see on a histological sample, and it contains both thick and thin filaments. And we can see that here contains a lot of the thick filaments, but you can also see the thin filaments shown here. Next up is the H band, and the H band runs along the midsection here of the A band, and it is going to contain only thick filaments, and it's only evident when the muscle is at rest. So when it contracts and it's pulled together here, remember the sarcomere is the contractile unit, we won't see that H band anymore. Before I talk about the M line, remembering back a couple of slides ago, we said that this is a myofibril, which contains various myofilaments. And then we have a bunch of myofibrils that are going to make up our muscle fiber or our muscle cell. The M line is going to be this protein that you can sort of see here that's going to stabilize these thick filaments, making up that myofibril. Next up is the I band. The I band is shown here, and it would be shown here as well. And this is going to be the lighter zone that we would see microscopically. It contains those Z discs that I mentioned earlier, and it's composed as well of thin filaments. And we can see the pink filaments here that we know are the thin filaments. You can also see the I band in this image, again, the lighter component. The Z discs, which I brought up before, these are going to extend across the midline of each I band, each light I band. So if this is the I band here, we know the Z disc is in the central part, and we know that it's the boundary between adjacent sarcomeres. And we know that because from one Z disc to another Z disc is going to be one sarcomere. And then similarly, from one Z disc to another one over here would be another sarcomere. This here is one sarcomere from Z disc to Z disc. And then finally, T-tubules, or those transverse tubules, which we'll look at in more detail soon, they would be located between the A and the I bands. So we're looking at right around here, we'd find these tubules that sort of traverse down into the muscle cell in order to allow the electrical current to be carried throughout. But we'll come back to that. We already know that there are going to be numerous nuclei that are found within muscle cells, but there's also a lot of mitochondria, which we know are the powerhouse of the cell, producing ATP. And you can see so many mitochondria scattered throughout. So to orient yourself, this would be a muscle cell here. And within this muscle cell or this muscle fiber, we have these myofibrils. So that's one myofibril. And then within the myofibril, we have myofilaments, our thin and thick filaments. The sarcolemma would be the plasma membrane of the muscle fiber. So think of the plasma membrane that we spoke about with normal cells or the common cells. This here is specifically reserved, this term, for muscle cells. So this is going to be what surrounds the entire cell or what surrounds the entire muscle fiber. The sarcoplasmic reticulum, we know that this structure here is the version of the smooth endoplasmic reticulum that we've talked about in the past. These sacs contain calcium, which is required for the muscle contraction, as we know. Remember the T or transverse tubules I had shown you on the previous slide, that they run in between the A and the I band. We'll just look back at this for a moment. So remember, they are located between the A and I bands. So right around this region. But now we can see them better in this image. These red tubules, they're not blood vessels, even though they kind of look that way. And we know that they're going to allow for electrical impulses to really travel through deeper into the cell. Remember at the start of the lecture, I had mentioned the term dyad. Di means two. Well, skeletal muscle possesses triads. Tri means three. This is going to be including one T-tubule and two sarcoplasmic reticulum sacs. So what we have here is a triad. You can see the sarcoplasmic reticulum, part of it running alongside the T-tubule. So the three of these together is called a triad. And what we'll see happens is when the electrical impulses run through the T-tubule, it's going to stimulate the sarcoplasmic reticulum to release calcium. We know that calcium is required for a muscle contraction, but how exactly does it cause a muscle contraction? When the muscle is relaxed, calcium is just going to be stored in sacs found within the sarcoplasmic reticulum. So to orient yourself in this image, we have parts of the, or sacs of the sarcoplasmic reticulum here, and we have the T-tubule extending in between them. So this would be a triad. And you can see the calcium is represented by these green triangles, and in this region here, and this region here, and here, there's a whole bunch of calcium that's sequestered together, and that's called calsequestrin. Think of it as calcium being sequestered together, concentrated calcium. And it's located near the calcium channels. It's almost like people lining up to leave somewhere together once the doors open. These channels here are voltage-gated channels, and they will open once they've been stimulated by a voltage or an impulse, which would travel down here to stimulate them. They would open, and then once they open, calcium will move just passively down its concentration gradient from where it's in high concentration inside the sarcoplasmic reticulum to where it's in lower concentration outside of it. So if we look at this image here, we can see that an impulse is traveling, it's moving through the T-tubule, and as it does, it's stimulating these voltage-gated channels. So this in red is the voltage to open, allowing calcium to leave where it's been sequestered by calsequestrin. It's passively moving out of these gates down its concentration gradient, and you can see it's now outside of the of the sarcoplasmic reticulum. Now, once we are done with the contraction, we want to reuptake this calcium to, one, stop the contraction, but two, use it for later. And so it's going to be transported back into the sarcoplasmic reticulum, and this is done actively. So we need ATP in order to pump it back in. So you can see a calcium pump here, and ATP is being used to pump calcium back into the sarcoplasmic reticulum where it originated. And so everything I just stated there and showed you is also outlined in this flow chart. Okay, let's back up a little bit and look at the nerve connection to a muscle fiber. So here's a muscle fiber. This muscle fiber we know is filled with, the muscle cell is filled with myofibrils, which are filled with myofilaments, those thick and thin filaments, which you can see the pink and blue shown here. This here is a nerve. More specifically, it's a motor nerve or a motor neuron. A neuron is a nerve cell. And we'll learn more about this in the nervous system lecture, but you can see that this motor neuron, motor referring to movement, is going to what we call innervate or supply, you could say, this muscle fiber. And where it connects with the muscle fiber is called the motor end plate, specifically on that sarcolemma, that plasma membrane of the muscle cell. Now, in between the motor neuron and the muscle fiber, there's going to be a gap here. A little harder to visualize on this slide, but this gap is called the neuromuscular junction, the junction or connection between a nerve and a muscle. It's also called a synapse, but it's a gap. And if we look at the motor neuron here, we can see these vesicles, and these vesicles are filled with a chemical called acetylcholine. And acetylcholine is going to travel to the end of this motor neuron here. It's going to release its contents. Those acetylcholine molecules are going to travel across that neuromuscular junction or that gap, and they're going to bind to acetylcholine receptor sites that are found on the muscle cell's membrane. And this event is really what will allow for a muscle contraction to occur. So if somebody has damage to a nerve that supplies a muscle, then we're not getting that communication to the muscle and it won't be able to contract. Okay, let's summarize. What we have here is a muscle fiber that contains myofibrils, and those myofibrils contain the myofilaments. So here we have that motor neuron, and we have that neuromuscular junction. So we're going to have a nerve impulse that reaches the end of the motor neuron, which is going to stimulate acetylcholine to be released from these vesicles. They will cross that synapse and attach to acetylcholine receptors that are found on the sarcolemma of the muscle cell. This will then allow sort of like a bit of a relay where now the baton is being passed on to the muscle cell, and that voltage will travel along the sarcolemma of the muscle cell. And what you can see happening when this occurs is that voltage is traveling down the T tubules, and that will stimulate the sarcoplasmic reticulum to release its calcium, and then calcium is going to initiate the contraction, which we will talk about next. So that calcium that's released from the sarcoplasmic reticulum that ends up in really the cytoplasm, which is the sarcoplasm of the muscle cell, is going to bind to troponin. And remember, troponin is a protein that's found on actin. And what it's doing is it's holding that tropomyosin in place, which is blocking those active sites on actin. And this will be occurring during relaxation. But what calcium is going to do is it's going to bind to troponin. So calcium is binding to troponin. This will cause troponin to activate and move tropomyosin away from those active binding sites. This will then allow myosin, part of the thick filament, to reach up its head and attach to that active site on actin. This process is energized by ATP. So it's like there's this stored ATP that's just waiting to be used. In this image here, you can see it actually even quite nicely where we have tropomyosin that's blocking the active sites on actin, and then when calcium binds to troponin, causing tropomyosin to move, so here's tropomyosin that has moved, now the binding sites, which would be called a myosin binding site found on actin, now it allows myosin to sort of reach up and bind to that site, which will initiate the contraction. So here we can see that energized myosin head that's reached up, and it's going to, and it's not just one of them, they're all going to be doing this as part of that muscle, is going to be reaching up and pulling in, shortening the muscle. And this process is going to happen repeatedly, so it's going to reach up and pull, then reach up and pull, and that will shorten the entire muscle fiber. What I just spoke about is sometimes referred to as the sliding filament, referring to the myofilaments, the sliding filament model of skeletal muscle contraction. In this relaxed state, we can see this is one sarcomere from Z disc to Z disc. This is another. You can see it's made up of the thick and thin filaments. You can see those myosin heads here. When the muscle contracts, you can see that the sarcomere has shortened from Z disc to Z disc, is now a smaller space. And we can see that a little bit in this image too. Now, looking at this simplified drawing where humans here are acting like the myosin heads, where they're reaching up and they're going to be pulling the actin filament, pulling them towards each other, which effectively shortens the distance between each Z disc. Then after they've done one pull, they will let go and they will move on to the next active site and pull again. So after the muscle contraction is done, it must relax, and this is going to mean that calcium needs to be removed from where it is bound to troponin and pumped back into the sarcoplasmic reticulum. So removal of calcium from troponin will cause tropomyosin to cover those active sites again. So they return to their original position. And because those sites are blocked now, myosin can no longer bind to actin, so it stops the contraction. And as we already know, calcium will be actively pumped back into the sarcoplasmic reticulum. So we know that ATP is required for muscle contraction. Remember that myosin head needs energy in order to reach up and grab onto actin. But where does it come from? Well, in order to use the energy from ATP that's stored in ATP, it has to undergo hydrolysis. Hydrolysis, remember, is the chemical breakdown of a compound due to the reaction with water, so the addition of water. So let's go over to this image here. Here's ATP, adenosine triphosphate, which has an adenosine and triphosphate, 3 phosphates that have a high energy bond connecting them together. In order to utilize the energy that's stored within ATP, what we need to do is undergo hydrolysis, and the energy can be released, but what we're left with is ADP, adenosine diphosphate. We only have enough stored ATP for about 2 to 4 seconds worth of a contraction, so where do we get ATP from? Well, the first thing I wanted to mention is that ATP is constantly being made from the breakdown of creatine phosphate. Creatine phosphate found in muscles is really nice because it's going to donate one of its phosphates to the ADP so that we can make ATP. In addition, we can also make ATP from glucose. We know that glucose, as part of cellular metabolism processes, will contribute to the formation of glucose. We also have glycogen that's stored in our muscles, which can break down and release glucose. Continued and efficient nutrient catabolism by our muscle fibers or our muscle cells are going to require 2 essential ingredients, and that's glucose and oxygen. Now, when oxygen is available, we would produce energy through the aerobic pathway. Aerobic is referring to using oxygen. But if oxygen's been depleted and we don't have as much, then it would be the anaerobic pathway. An is lack of, so lack of oxygen. Now, the aerobic pathway is a catabolic process that's going to produce the maximum amount of energy available, energy as in ATP, from each glucose molecule. And it's going to be using oxygen to do this. However, if oxygen concentration is low, then our muscle fibers will shift towards using another catabolic process that we call anaerobic metabolism, or an anaerobic pathway. The process of producing ATP through the aerobic pathway is a slower process, but it will supply energy for the long term. The benefits to an anaerobic pathway is it is very fast, and it's going to be what supplies energy during your first few minutes of maximal exercise. And we'll look at that a little bit closer in this chart here. But before that, I wanted to look at this image above, or this graph above, to show you that the aerobic pathway shown in red is going to supply energy for muscle contraction for a longer time than the anaerobic pathway, which we can see here, with energy on the y-axis and time on the x-axis. Now, if we look at this chart at the bottom, what we can see is that when we're looking at 10, 30, and 60 seconds into exercise, we're primarily using our anaerobic pathway compared to our aerobic pathway. But then once we start getting into minutes of exercise, we start seeing a bit of a shift occurring. And by the time we've reached an hour of exercise, we're using primarily our aerobic pathway to produce ATP. Again, we're going to be getting much more ATP through the aerobic pathway than we are the anaerobic pathway. Okay, moving on a little bit to a slightly different topic, looking at the function of skeletal muscle organs. Now, although each of your skeletal muscle fibers are going to be distinct from all other fibers, they still operate as part of a large group of fibers that form that skeletal muscle organ. And skeletal muscle organs that we will just often simply call a muscle are going to be composed of a bundle upon bundle of muscle fibers that we know are held together by a fibrous tissue. And we talked about this anatomy already during one of our other lectures. Remember that each muscle fiber receives its stimulus from a motor neuron or a motor nerve. And if we take one of these motor neurons plus the muscle fibers that it attaches to, that's going to constitute a functional unit called a motor unit. So a motor unit is the motor neuron and the muscle fibers that it innervates. So what we can see as an example here are 3 different motor units where we have the motor neuron here and the multiple muscle fibers that it innervates. So that's one motor unit. We have another motor unit and a third in this simplified example. And the number of muscle fibers that are found in each motor unit is going to vary. So if we need very precise small movements, our motor units, so the motor neuron and the muscle fibers, are going to contain overall few muscle fibers. But when it comes to more powerful, stronger movements, this is where we're going to need a motor unit that consists of a motor neuron and many muscle fibers. So the difference here would be using your hand to do something versus helping somebody move something. And here again is a motor neuron and the multiple muscle fibers that it innervates, making up a motor unit. Moving on now to the topic of muscle tone. Muscle tone, also known as tonic contraction, is going to be this continual partial contraction within a muscle. And it's really just a low-level continuous contraction that's going to be characteristic of how your muscles are when you're just awake. We maintain muscle tone because of negative feedback mechanisms that exist and are centered within our nervous system, specifically within the spinal cord. Now, at any one moment, a small number of your total fibers in a muscle will contract and produce what's called a tautness of the muscle rather than a recognizable contraction or movement. Now, a muscle that is going to possess less than normal tone is described as flaccid, and muscles that have more than normal muscle tone are described as being spastic. Next up is the stretch reflex, and the purpose of the stretch reflex is to maintain a relatively constant length as load is increased up to a maximal sustainable level. So what does that mean? Well, in this image here, we can see a person that's carrying a box. Then another box is added to it, which increases the load. So the box would be the added variable. Muscle length is going to be automatically increased. Now, a stretch is detected by muscle spindles, and this is really a sensory signal that's sent through a sensory pathway where this info is then going to be integrated in our spinal cord. And the decision that we need is to increase contraction in order to manage this increase in load. And so this message is going to be sent along motor nerve or a motor pathway in order to signal the muscle to contract to manage that increased load. And this is what brings us back to homeostasis. So it's a negative feedback mechanism. And of course, it only will occur up to a maximum sustainable level. So in other words, if it's too heavy and our muscles can't handle it, then we won't be able to continue holding on to that load. We're going to look now at the difference between an isotonic and an isometric contraction. An isotonic contraction, iso meaning the same and tonic referring to tone or tension, an isotonic contraction is a contraction in which the tone or tension within the muscle remains the same, as I've just stated, while the length of the muscle changes. There are two varieties of isotonic contractions. We have concentric and eccentric. Concentric contractions are those in which the movement is going to result in an actual shortening of the muscle. So for example, if you were to pick something up, that would be a concentric contraction. If you lifted up a weight, that's a concentric contraction. So the muscle is shortening. With an eccentric contraction, these are when the movement results in lengthening of the muscle that's being contracted. So you pick up a weight, that's a concentric contraction, but then you lower it back down, that's an eccentric contraction. So in any case, you're using the same muscle, but one is causing shortening and the other is causing lengthening. And with this, we are going to be using energy to pull for the myosin or the thick filaments to pull on the thin filaments, which is going to change the length of the fiber's sarcomere from Z disc to Z disc. Isometric contractions, in contrast, this is a contraction where the muscle length is going to be or remain the same while the muscle tension now increases. So metric has to do with, say, the measurement or the length of the muscle is remaining the same. Now, an isometric contraction is not going to be actually moving anything, but let's say you were to hold on to something, even hold on to a weight, but not actually move your arm, that would be an isometric contraction. So there is an increase in muscle tone, and you can even feel that muscle tone increasing, but the length of the muscle has remained the same. Most body movements are going to be occurring really because both types of contractions are occurring. And finally, there are different conditions that can affect the strength of a muscle contraction. So moving clockwise here at the top, the question could be, have you been lifting already for a while and fatigue has set in? Trepe referring to the continued muscle contraction. Another question would be, how many muscle fibers are actually activated? The more that are activated, the stronger the contraction. Is the length partially contracted or partially relaxed? What is the load that we're lifting? And then the amount of load. So is it too heavy? Your body will know this and it will either initially adapt to it through that stretch reflex, but it will also force you to abandon the load if it's too heavy to avoid damage to your muscles. That is the end of today's lecture. Thanks a lot for your time. Bye for now.